Thank you very much. We've now completed our presentations by the panelists, and we're open for questions. So please head up to the microphones. While people are doing that, I'll start with the first question. We have really, we heard from the group that it really is going to take a public-private partnership to tackle some of these issues. And Rick, I know I've talked to you about this before. I thought I heard you mention the idea that we might study multiple drugs in class in the same clinical trial, with companies bringing their drugs to bear. Can you describe that a little bit more, and then I'd like to have some reaction from Mace and Bill.
One of the things that we were thinking of, obviously there are common development patterns that we see, and it's very interesting when you take a look from pharmaceutical company from afar, because obviously they want to reduce risk, and there's nothing like a successful drug and then to pattern other drugs around that successful drug. So for example, the ALK inhibitors, we now have, I think, 4+ sons of crizotinib, so to speak, coming up, each with the same development pattern, because here again, it's about minimizing risk. So they're all studying their drugs in crizotinib-resistant disease on a single-arm trial and looking at response rate. And then that's for accelerated approval. Then they want to go up front and compare themselves to crizotinib. Well, that's going to be very difficult with all of these drugs, and one could question what's even the benefit of looking at that. And then you get the issue that Mace even brought up, should you even be using these drugs sequentially rather than saying one is the winner, so to speak. But given that, one could envision, for example, a trial where you had a common control arm, crizotinib, and then 4 arms being the other drugs and studied simultaneously. You would use a common control arm, for example, and therefore cut down a large number of patients that would be needed in an independent randomized trial. You would have greater utilization of resources. From a regulatory point of view, we do not have a comparative efficacy standard. In other words, one drug doesn't have to be better than the other new drug, so to speak. The statistical plan would be looking at each new drug comparing itself to crizotinib, so to speak. So there would be no ultimate winner. All of these drugs could be winners if they showed that they were better than crizotinib on the basis of, for example, response rate or time to progression, whatever that endpoint would be.
So, Bill?
So that's a way of looking at trying to leverage, newer drugs coming out and trying to have, because I think it's going to be very difficult to have all of these drugs do their own independent trial. Ultimately, however, even though there will be multiple winners here, there will be comparisons, not necessarily done by the regulatory agency, but by investigators, et cetera, that will look at this data and be able to say, boy, this drug A looks better than any of the others that we tested against crizotinib. So that is a danger, but not a regulatory danger. But I think the American public really has the right to know that information.
What would J&J think about that?
No, it sounds like it's a very good idea. In fact, we as a company, we are participating in Ellen Siegel's effort with friends and with the FDA for non-small cell lung cancer. So it makes very, very good sense from all sorts of issues, including the equipoise issue that Rick mentioned earlier. The other piece that it probably helps get around very nicely is the European issue of the fact that a drug gets regulatory approval has no direct influence, except if it's not approved, on getting the drug reimbursed. So the standards for reimbursement in many European countries are clinical benefit and comparative effectiveness. We even have heard recently with a diabetic drug that, well, if your drug is 25% better than metformin, which costs almost nothing, we will pay you 25% more than we pay for metformin. This is the kind of thing that puts a huge damper on the drug development process. So this type of trial, I really think, helps solve several of those problems.
So Bill, I really liked your second point about finding surrogate endpoints too, and that of course requires really truly understanding the mechanism of action of the drug and how it would intervene with precursors to the disease. But let's take a step back and compare with when you're profiling the disease. One of the big challenges, of course, we're having with clinical trials is it's very hard to find patients to match a particular indication. So we talked about, a patient with a hedgehog pathway defect. How do you find that patient? Well, should a pharmaceutical company have to sponsor the molecular profiling of every patient across multiple sites, or can we create a clearinghouse where that information is made available so patients can be recruited? Well, perhaps the same idea could apply to prevention. One of the things that we've been doing with pediatric hospitals is thinking about sequencing children early for disease risk identification. So you can imagine every child walking through a hospital would get whole genome sequencing in the not too distant future. That information would live with that child through their health care, and if there are risks identified, you would prevent. The problem is we don't have those interventions. And so one could imagine creating a similar type of germline genome clearinghouse where if there were interventions, you can match a patient to go on a prevention trial, and provided you had those types of early surrogate endpoints, you could turn those around quicker. But did you want to expand on that? Is that something that you were thinking about?
Thanks for the question. One of the critical things that I believe is happening, and many people in the room know more about it in detail than I do, is that going from predictive algorithms that we currently have that give relative risks that are not really necessarily actionable, because you wouldn't want to treat every patient in the world if their relative risk is 1.2. As those algorithms, as more data from longitudinal data sets become available, the algorithms will get better and better, and the susceptibilities become, or the prediction of susceptibility or the prediction of risk become more robust. Then I think you have a better chance of making an intervention that makes sense. The issue that you raise is validating the surrogate endpoints. And by definition, I believe that validation will take a very long time. So whenever someone says, in my company, that's going to take a very long time, I always said, yes, so that's why we need to get started right away. And doing it through consortia in some logical way with colleagues at the agency to really start thinking about, well, how would we validate a surrogate? I think that's exactly what we need to do.
Nancy?
Nancy Davidson, University of Pittsburgh. I want to ask something that is certainly out in the medical press and in the lay press, and which we haven't really talked about, which is the concept that we're accused of overdiagnosing and therefore overtreating, that there are a lot of malignancies that may not actually threaten that person during their natural lifespan. How are we thinking about that? How are you thinking about it, panelists?
Do you want to take the first crack at that? It's a tough one, I know.
I'm probably not the best person because I've been treating patients with metastatic disease, but I want to emphasize again that I think that bringing these diagnostics and therapeutics to patients with established disease but not highly metastatic is a sweet spot that we've barely touched, if at all, in the solid tumors, and may be much easier to address than the prevention aspects, which ultimately are going to be hugely important, but I think have some real challenges as far as addressing right now.
Jim, I know you have some trials for early disease, or Liz?
Well, I was going to mention from the diagnostic regulatory side, that's something we worry about all the time is, is that really a lesion that matters, or is that something that just rides along? And in fact, the more powerful technology gets and the less upfront information you need to have, you can see the whole genome and look for what you think might be wrong in it, the more critical that problem becomes, because really nobody's the same and you have to sift out what's just normal variation from what's really a problem. And I actually don't know any efficient way to do that other than to gather people with similar changes and look to see if there's a difference among them. But so far, people are not doing that on a broad scale.
Don't you think that although this is a practice of medicine type of question and not a regulatory question as such, don't you think it really revolves around how effective the therapy is that you're introducing? What are the side effects that you are putting on that patient, so to speak? So the issue here is, even if you had this wait and watch type of issue, for example, in follicular lymphoma, here again, if you had a therapy that cured those patients, then it's a much different thing than whether you're just having a mild perturbation, so to speak, of the natural history of the disease, and they're going to come back later with the disease, and you're really not altering the eventual outcome of the disease. So I really think it's how effective the therapy is that you're introducing.
Dr. Calfano?
So it's a question for Elizabeth Mansfield. So I've heard some concern or maybe perplexity about how sort of system biology can be sort of introduced in the context of regulatory study. And I wanted to understand this a little bit better, because obviously we think of system biology more as an approach to understand the disease so that you can then come up with a relatively small number of multiplex panels that literally include anywhere from 2 to 3 to 4 genes that can then be used to associate particular response to a drug, et cetera, as opposed to actually really running sort of IND certification and studies for approval of new drugs based on some complicated formulas and models. So maybe if you could give us a better sense of where the concern comes from, it would be really helpful.
So I have no concern with systems biology as knowledge generator at all. I think it's fantastic. What I would be concerned about is tying systems biology to therapy prematurely, to think that you know more than you know on the basis of systematic knowledge that's incomplete or may have pieces that are incorrect. So that I have no problem whatsoever with systems biology. It's just that from a regulatory point of view, I feel that we need to have something better than, and I hesitate to use this word because it sounds terrible, I don't want people to be experimenting necessarily on the basis of systems biology that lacks a lot of biological knowledge yet. As I mentioned, some of the things that we don't know, very simple things that we don't know, I think we still don't know them. So making the leap to trying to put together a lot of pathways and so on just piles on top of that, things that we don't know.
Bill, you had a comment?
I think one of the really important things that was brought up today about the N of 1 trials, and no matter how sophisticated we're getting, and we certainly are, in the real world, in the milieu of the different genetic abnormalities in cancer, when you make an intervention, there are 3 possible outcomes. It could improve things, it could make things no different, or it could make things worse. And the most critical thing is we capture the data. And I think the movement to do that in an organized way is a very important first step. The derivative, which might even be more important but much more difficult, is to understand when these drugs actually don't work or make things worse, despite having what would have been thought as the correct actionable mutation, deletion, translocation, whatever it is. Knowing why it didn't work would be even better, but much more difficult.
And yes, I agree, and nobody looks at that very much. Why doesn't it work? The other risk, of course, is not only that you get a therapy that makes you worse, or is that you might pass up something that would make you better in exchange for something that either does nothing or makes you worse. So there's a lot of threads to tie together.
Bill?
Bill, I love the lenalidomide example for several reasons. First is that, if I'm not mistaken, the repurposing of the thalidomide and lenalidomide for myeloma certainly involved a lot of important legwork done by academic investigators who had access to the compound. Secondly, my colleague Ben Ebert and I have papers in press that show the mechanism of action finally for lenalidomide. And it turns out when lenalidomide binds to a particular ubiquitin ligase, that ligase can now target for destruction 2 transcription factors that are critical for myeloma cell maintenance. And so it is a targeted agent. And I only raise that point because I think we've talked a lot today about going from for example, mutations to what the response is going to look like in the clinic. But I think you'll agree there's still room for going from responses in the clinic back to the laboratory. And some of the more exciting uses of genomics I have seen have been analyzing some of these extraordinary responders in clinical trials and figuring out why, in hindsight, they had these extraordinary responses. And I think it's going to be equally important, if not more important, moving forward.
Great. Rick?
Rizal?
Could I just ask Liz a question about the systems biology issue? I think that increasingly we realize because of the complexity of many of these tumors that we're going to have some way to integrate the information, and not being able to integrate it really hobbles us because we have to guess at single mutations, which one are we going to approach. So what would be the path to having a systems biology approach that is tenable? Because my own concern is that it's worse not to be trying it than be trying it and perhaps have it not be perfect at the beginning.
Yeah, so I think for almost anything you want to do that's reasonably safe, there's a path. So, you know, it's not something we've done a lot before, probably more in imaging than in other types of diagnostics. But I agree that it's maybe better to try it than to not try it. In some cases, you do still have to, from the regulatory side, look at the benefit risk. And I think in that case, we also have to take into account, to some degree, patient preferences. Are you willing to take a risk on this? Or, you know, are you willing to enroll in a trial on this? Just so that we can get an idea that it doesn't have to be perfect, but it works better than what we have. And I mean, in the diagnostics world, nothing is perfect. We know that. No test is perfect. And we don't expect them to be. And some of them, in fact, are pretty darn not perfect, but they're still better than doing nothing. So I think there are paths forward. It would take some discussion because it's actually an extremely complex system, obviously, even to work out, much less to ask the right questions about if you don't know much about it. But I would say, you know, interaction among stakeholders would be the place to start to see, you know, what we actually need to do in order to introduce this.
Yes, please.
So I just wanted to follow up with that. I think that's a really good idea. The idea is that if you think about patients who have failed everything else, and usually the last recourse is a phase 1 trial, the success rate is under 10%. And I think that might represent an opportunity where the risk is very high, and if there's nothing left, then we can use very sophisticated systems approaches, looking at pathway convergence and so forth, to get a little bit more intelligent about putting patients on phase 1 trials where they're going to benefit and get beyond the 4 to 10% success rates up to the 20, 30, 40%. And Dan Von Hoff has shown that. He's shown that you can get up to 30% response rates on phase 1 if you use molecular intelligence. So I think that might represent one of those opportunities.
We had an online comment from Kate Dobson who wrote in reminding us that many of these patients go on clinical trials in the community and are treated by community oncologists. And there wasn't really a question there, but I actually have one, and more of a practical aspect, and I'll start with Jim. You know, looking at policy issues, how available are some of these tests to patients in the community, to diverse populations, and you know, what are efforts that NCI, for example, might be doing to help that?
So, excuse me, in all of these larger trials that for which NCI is supporting the bill for the molecular characterization, they'll be done across our entire network. So 3,000 sites, including the CCOPs, et cetera. That's really about as good as we can do. But I think because you have to involve people who know how to do clinical trials, but it will bring that testing at no cost in a research setting to many, many patients around the country.
I know you work in your company with an ALK inhibitor, and you're looking for an ALK rearrangement, which isn't the easiest test to do. We've had several controversial results at our tumor boards. How do you feel the community is doing getting access to those tests and getting the patients in that might benefit from your drugs?
That's an excellent question, because I think if you surveyed 100 oncologists, 99 out of the 100 would be aware of ALK-positive non-small cell lung cancer. When you look at the percentage of patients with non-small cell lung cancer who are tested for ALK, that falls down to about 65%. So even today, we're not seeing 100% testing. That goes for EGFR and goes for other molecular abnormalities. So I think one of the biggest challenges is not awareness, it's access. It's the obstacles that stand in a busy clinician's way to get to identify where the test is available, how do you order it, when will results be back, will they be back in a timely fashion for the therapeutic decision that they're facing? Because that's also a problem that we've uncovered. That is, there are laboratories around the world that will do sequential testing. So for a newly diagnosed non-small cell lung cancer patient, they'll test for EGFR. If it's negative, then they'll test for ALK. Meanwhile, what that introduces is a delay in the diagnosis. Meanwhile, the patient is waiting for a treatment decision to be made and treatment to start because they're usually symptomatic. And the physician is having to wait and becoming increasingly uncomfortable. They're going to intervene with chemotherapy and then use that information only after the chemotherapy fails. And we know that that may not be the right decision for EGFR and maybe not for ALK patients either. So I think that this is one of the good examples of how we have to recognize not just the direct question in front of us, getting the patient tested, but the obstacles that may not be so apparent to getting that test result, getting the test paid for and done on a routine basis.
Hi, Lily Shockney from Johns Hopkins. I have a couple comments. One is that in looking at a growing increase in the shortage of oncology specialists, so that more and more docs are going to be taking care of, or less and less docs are going to be taking care of more and more patients, there's going to be a smaller window of opportunity to be holding that conversation about enrolling in a clinical trial. We also have the issue of we're all relying on the pathology to be accurate. You know, the pathology holds all the cards, and not all pathology is accurate. We've recently looked at our second opinions for pancreatic cancer and found that 22% didn't have pancreatic cancer. That's not good. And not everybody comes anywhere for a second opinion. With the issue of bringing stakeholders together, I think one of the most important individuals that should be there are patient advocates, so that stakeholders that are competitors to one another can be reminded by patient advocates who probably were patients themselves why they're in the business that they're in. So.
Good points. Any comments from the panel? Lowell?
Probably a question most directed to Mace and Bill, but to anybody on the panel. Over the past year and a half, the Cancer Research Committee at ASCO undertook an effort to try to define, looking to the future, clinically meaningful outcomes in clinical research. It proved to be a rather interesting set of argumentative meetings because there was really a stretch at getting any consensus among those thinking about metastatic breast cancer, colorectal cancer, non-small cell lung cancer, and pancreas cancer. And on the one side are those who recognize the realities of incremental gains in many of these diseases, although we're all aware of extremely small incremental gains in some of them, and others who felt the impatience of the scientific and clinical communities spurred on by the advocate community for more progress faster. And I guess I'm wondering how those leaders in industry or clinical investigators like yourself, Roy, or Rizal, are thinking about this. In other words, are the endpoints that we're shooting for appropriate in the era of targeted therapy? Should we be considerably more ambitious, raising the bar, rejecting that many more, even though I know many compounds never make it, but should we really be raising the bar, or are we doing the best we can?
So actually, I think Roy and I sit on that committee, and so we're involved in these, as you do, Lowell. And so I think that the message that was conveyed by this effort is embraced by everyone, and that is that we should be setting the bar higher for what we expect from new therapies, for them to be meaningful to patients and demonstrate the value that justifies the price that's asked. I think that having said that, how do you tie this concept in to the actual pragmatic approach to new drug development, because each group was asked to identify what they considered a meaningful improvement. And I don't know if it was established or they all ended up in the same place, but they defined that in terms of overall survival. Now, for certain cancers like pancreatic cancer, you could use overall survival as your primary endpoint for clinical development, and it will get you there pretty quickly, unfortunately. But for many other diseases, and some include now breast cancer, lung cancer, colorectal cancer, you're actually going to, if you use that as the measure for drug development approval, you'll actually slow down the pace of new drug development. And as Rick and I know from our previous lives, the doubling in survival in advanced colorectal cancer occurred through a series of small incremental steps, none of which would probably have made the criteria for what this subcommittee has set out for adequate improvement. So what I've tried to articulate during those committee meetings is that we don't want this aspirational document to be an impediment to that incremental approval. I think every investigator, every company, every drug sets out with the idea that it's really going to transform the treatment of a particular disease, and very often come up short to that goal. But whether not achieving that audacious goal is equivalent to failure, I think, is the subject of question. And then it's really an issue of how we as companies and investigators and regulators and payers view that improvement as being meaningful to the patient. So I think we have to keep all those perspectives in mind when we think about where we want to set the goals for new drug development.
So I'm also on the committee, but I'm not burdened by good attendance, so I can say whatever I. So, as many of you know, in the UK, they've solved the problem for us, sort of, by creating by NICE, National Institute of Comparative Effectiveness, or something like that, which looks at qualities. What is the cost for a productive year of life gained? And they set it at a certain threshold, even for oncology drugs, somewhere around 50,000, 60,000 pounds, something like that, maybe less. And if you don't make it over that threshold, you don't have reimbursement for your drug. So I'm not advocating that is the way to do it, but it is a way. And if you go look back, it would, at the drugs that have been approved and not approved in that system, you could really scratch your head sometimes. But as a general rule, this concept of your drug improving quality of life for X many years at certain amount of societal cost isn't a bad place to start. And perhaps some better derivative of that is worth discussing.
Yeah, I certainly think in this molecular age we need to shoot higher, realizing that there are going to be drugs that will have incremental benefit. One thing we haven't discussed much in this panel, and we're going to have to end, is quality of life and patient-reported outcomes, which are going to be essential for all of these studies, and maybe we'll hear more about that this afternoon. But it was a laudable effort. Okay, I think we're pretty much at time. I want to thank all the panelists for a very exciting discussion. You've given us plenty of issues to think about for future meetings. And now what we're going to do is we're going to go outside and we're going to pick up lunch, and we're going to come right back in, because Dr. Mendelsohn is going to be sharing the keynote presentations, and I think he has a few words on that.
We don't have to come back in.
Yeah.
Time to.
We have time to relax for a few minutes. We'll be back at 12:15. So.